Good morning, everyone. And first, I would like to sincerely thank the organizing committee of this International Symposium on Artemisia for inviting me to be part of this roundtable. I would like to give a special thanks to Dr. Arnold, who has been in connection with me from the beginning. I also want to apologize for not being in Tanzania in person, but thankfully, we have the opportunity to join remotely to be part of these discussions. Okay. Well, my talk reflects the 30 years of work we have done here in the Federal University of Technology here in Paraná State. And from the beginning, we start to work with only with physiology of Artemisia, and after we worked with agrotechnology, and now we are trying to test elite genotypes of Artemisia against some disease in humans or animals. Also, I would like to introduce to you my team. These people, they are from the biochemistry and plant physiology lab team. I have here people from that are working with me from 2023 2000 to 2025. The main work now is doing by Lucas Dalla Corte. Lucas Dalla Corte is working in his PhD with selection and breeding of Artemisia annua, trying to reach these elite Artemisia annua genotypes with high quantity of artemisinin. Bruno Fontoura, he's working with nanotechnology. He's doing nanoparticles with Artemisia annua extracts, and it is part of his PhD thesis. Another person who is working with Artemisia is Amanda. Amanda is doing her residence right now. She's veterinary, but she was testing Artemisia annua extracts for control to control endoparasites in chips. Here's some of our papers exclusively with Artemisia. We work with another plants, another medicinal plants as Varronia curasavica, and also with commodities as soybean and maize. And here we have the global demand for artemisinin and the derivatives. We can see still growing. It's a bit old, this data, but it's from 2021, okay? And who are working with Artemisia for a long time, we remember when the price of the artemisinin, it was around $1,000 per kilogram. And Sanofi tried to produce a synthetic artemisinin with a better price. The cost of this artemisinin, this synthetic artemisinin, it was around $400 a kilogram. And the producers of Artemisia, they start to put down, the producer of artemisinin, they started to put down this price. And the price today is around $150 and $200. We believe we still have a market for growers of Artemisia. And here we have an example. We have here in Brazil a company. It's a public company. They produce one kind of ACT. The name of the ACT is ASMQ. The company is Farmanguinhos Fiocruz. They produce around 1.2 million treatments per year. Sorry, it's not per year, but it's from 2008 to 2021. And we were expecting to sell artemisinin to this Brazilian company, Farmanguinhos. And we had this experience. The Brazilian government was encouraging the production of artemisinin and analogs in Brazil through laws. And this company, Cercetec Química, start to produce artemisinin in Brazil. But unfortunately, Farmanguinhos start to buy artemisinin and derivatives from India. it caused frustration, and we are still looking for another opportunities. We believe this symposium can be uh, a good place to discuss these new opportunities, including the animal area, the use of artemisinin or artemisia or derivatives to treat animals. But now let's talk about our work. Um, in the beginning, we start with many works trying to uh, understand the physiology of Artemisia, including if Artemisia is a C3 plant, C4 plant, regarding the behavior under different kind of photoperiod, etc. Let's show some of our work. Um, in that time, I was thinking about which kind of behavior Artemisia annua has or have regarding the photosynthetic physiology. We did some cuttings, and we saw some different. We found this vascular bundle sheet, and this is a special or a particularity of C4 plants. But when we use together histochemistry and carbon isotope composition, we could find that Artemisia annua is a C3 plant. As we know, C3 plants, they have a problem in hot places. And this problem we call photorespiration. The plant uh, the plant lose CO2 and don't do not produce the same quantity of biomass in warm places because the photorespiration. We can compare here two places in Brazil, Pato Branco, where we are, in the south of Brazil, and Campinas in São Paulo region. Okay, in Campinas, the production of leaves biomass is 2,000 kilograms per hectare, and in Pato Branco, 3,000 kilograms per hectare. The same quantity of days in the field, okay, the same content of artemisinin, but here in Pato Branco we produce 30% more of artemisinin than in comparison with Campinas. Okay, this is a kind of a problem when we want to grow artemisia near low or small latitudes. Some people will appear during my presentation, but it's to give honor to them because they were part of the work. Dr. Fernando Broeto, he was my advisor when I was doing my master in this subject. Another research we did, it was trying to give some stress to Artemisia. And we found good results with a moderated stress with a water potential around minus 1.39 megapascal. Artemisia annua produces 30% more artemisinin in comparison with the control, okay? And this is a good information. When we harvest Artemisia, we can regarding the temperature affecting the response to regarding the temperature affecting the flowering and some response together with photoperiod. What we saw in some experiments, we can see difference between genotypes. For example, one genotype from China, another one from Vietnam. Okay, they have a different number of photoinductive cycles necessary for a floral initiation. The Vietnamese needs around 30 days or 30 photoinductive cycles, and the Chinese one needs half of this quantity of cycles, around 14 cycles, photoinductive cycles. Okay, and also when we did the experiment in a hot season, what we saw that 100% of the Chinese plants flowered under short days. We know Artemisia annua is a short day plant. This This result was found by Ferreira, Dr. Ferreira. But what we saw in our experiment, some genotypes, as the Vietnamese, flourished less than the Chinese one. And we know, as a physiologist, that the temperature can alter the behavior under photoperiod, the plant behavior. And we repeat the experiment in a season with was less warm. The temperature, the climate was less warm. We saw with lower temperatures. Now only the Vietnamese genotype was inside the chambers, and we gave to the plants low temperatures in comparison with the first experiment. And what we saw, the most part of the plants flourished. And here we could see that the temperature also is important for the flourished of Artemisia. Regarding the best time of the day to harvest Artemisia, when I was in Israel, I made this experiment. And what we found, uh, just regarding artemisinin and dihydroartemisinic acid and artemisinic acid, regarding the content, we didn't find uh, results confirming 1 hour of the day, especially ideal to harvest Artemisia. We can find in many papers published, they used to say the morning is the better moment to harvest, but we didn't find it for Artemisia. During my time in Israel, these good people, they hosted me, Dr. Nativ Dudai, famous researcher in medicinal plants, and Dr. Olga Dudai, just to give honor to them. What about ontogeny? Here in Brazil, Artemisia annua used to flourish around February and March. Um, and the critical photoperiod is around 13 hours. And what we saw when we transplant the plants too late, near this period of uh, flowering, what we saw when we put the plants too near February and March, look the number of the days in the field, 31, 20, 28, 41, the concentration of artemisinin, the content, it's too low. Artemisia annua needs to be near 100 days in the field to reach the ideal content, near 1%. Let's talk about agrotechnology. This is the master thesis of now Dr. Marcus Palladini. His experiment showed to us if you put more, if the level of fertilization is increased, the plant will produce less essential oil and less artemisinin. Okay, what it means, the plant to produce more active compound, in this case artemisinin, needs to be stressed. You cannot give to the plant too much fertilization. Okay, but when we do not put enough quantity of fertilization, we have problem with the production of biomass. Okay, and when we are doing the productivity, the yield of artemisinin in kilograms per hectare, we cannot find difference between less fertilization and more fertilization. Okay. This is the result of the PhD thesis of Dr. Silvia Zamaria. She did the same. working with fertilization levels, and she found the same results that Palladini found in the field. She worked with a Hoagland solution, okay, and the complete Hoagland solution produces the complete Hoagland solution here produces less artemisinin, okay, but produce more biomass. And when you multiply the content by biomass, we will find more artemisinin. The productivity, the yield will be better with 100% of the Hoagland solution. Here we saw some gene expression with half of the Hoagland solution. She could find a better or more expression of some enzymes of the pathway of artemisinin biosynthesis. The plant density also affects the content of artemisinin in plants. This is the master thesis of now Dr. Diogo Capellin, and what he found. Uh, the maximum technical efficiency he found with around 80,000 plants per hectare. Okay, and why he found the best content of artemisinin with this high quantity of plants? Because when the density is bigger, you have less stems and more leaves. Okay, and the yield also it's around 80,000 plants per. The best yield of artemisinin is around 80,000 plants too. And of course, it will depend of the genotype. Okay. Let's talk about the breeding program, our breeding program of Artemisia. Okay, let me talk a bit about the Artemisia annua breeding program in Brazil, and also let me give honor to two very good friends, Dr. Pedro Magalhães and Dr. Ilio Montanari. They are the precursors. They start to do the breeding in Brazil. When they got the first samples of Artemisia, the first seeds, they found the content of around 0.01% of artemisinin in that plants and that genotypes. And during the 90s, they did a lot of work and they increased the content to 1% in the biomass, leaves biomass, and one production of around 25 kilograms of artemisinin per hectare. Okay, here we have some of the hybrids they developed. I know they did a work in agreement with Mediplan in Switzerland. Okay, but they got a very good result from 0.01% to 1% in the leaves biomass. And Dr. Magalhães, he got his retirement, and then in 2017 we did agreement between Unicamp, his university, and our university, the Federal Technological University of Paraná, and they gave to us the germoplasm, and we continue. We start to do breeding here in Pato Branco with materials from CPQBA, from Unicamp, and doing selection in the Unicamp germoplasm. We found the results with we found plants with 1.67% of artemisinin. And here we can see the first year of this work with the plants that came from a CPQBA Unicamp. Okay, we did a selection between and within families. Okay, from this population, that come from CPQBA. For the selection, we use agronomical, phytochemical, and physiological traits to do the selection. Here is the result of the first year. It was the master thesis of now Dr. Jorge Zanata. Zanata selected plants that reached 1.3% of artemisinin. And after his work, Lucas Dalla Corte started his master, and he found plants with 1.67% of artemisinin, showing progress in this kind of selection.
So, my name is Lucas. I am a doctorate student, and I work with Professor Abramo. So, first of all, I would like to talk about this article. It was published in 2018. It was a new approach about artemisinin accumulation in plants, and with the data of this article suggests there's a different chemotypes, someone with high artemisinic acid and high artemisinin, and these two compounds have a complementary pathways, while competing with artemisinic acid. And this article suggests crossing plants with. So now I'd like to talk about my doctorate project. It's called Selection of Superior Genotypes for Breeding Populations. This project starts in 2022 with 10 Artemisia genotypes from a collection established by George Ferreira at the USDA. And the correct collection was subsequently transferred to Brazil and is currently maintained in the university, at the university. This collection comprises Brazilian, Swiss, Vietnamese, and Chinese sections and genotypes, which have been cultivated in the field for evaluation and selection based on agronomic and phytochemical traits. Two of these genotypes they are presented in the collection had in the previous results showed almost 22% of artemisinin content. So new clones were prepared and cultivated in the field for evaluation and selection for agronomic traits and phytochemical in Brazil. So, with this information, we applied a multiple trait genotype idotype distance index. This is a plant breeding methodology who enables the simultaneous consideration of multiple traits, allowing the breeders to enhance specific traits while minimizing the others. By the integration of the agronomic and phytochemical data, by the agronomic data, we evaluate planetary mass, the number of grains. We identified four genotypes that are the best candidates for using a plant breeding program. But we have a problem with the genotypes we selected were 3N39, MDP7, 3N13, and C1 Purdue. But we have a problem with the MDP7, and we have a problem with its flowers, so we change for the MDP4, it's the next one on the list. So once the superior genotypes were selected, the cross-breeding program began. Flowering inducing of plants was the plants had flowering induced in a phytotron chamber, and at the first sign of a flower bud emergence, plants were closed together, the two plants to cross in organza bag to prevent uncontrolled pollination and fertilization. And after the flowering, indeed, the plants were separated again and bagged individually to collect seeds separately. After seed collection, they were sown in trays with substrate for germination and seedling establishment, and later transferred to the field. So, although the artemisinin contained the progeny was not higher than the other parents, we had a good result. Like 6 of the 15 genotypes plants we had were identified as superior. And a good point is among these six, Four genotypes were from the same genealogy, originating from the cross between genotype C1 Purdue and genotype AM3N13. And about this genotype, C1 Purdue is a genotype with a high artemisinin content, but also a high dihydroartemisinic acid and artemisinic acid content. And 3N13 is a genotype from selecting Brazilian plants which had an average of artemisinin content and a very low dihydro and artemisinic acid content. So this represents what was proposed in the early article. Crossing plants with a high artemisinin content and a high conversion ability, we can produce we produce superior genotypes in a plant breeding.
Okay. Um, we talked about some research with ecophysiology, agrotechnology, and selection and breeding of Artemisia annua to reach elite or elite genotypes. Okay, and after got these elite genotypes, we try to test them doing biological tests. Okay, with animals, human, and I will finish with some information regarding allelochemical effects of Artemisia. Okay. Um, okay, let me talk about the antiparasitic potential of this elite Artemisia in the control of gastrointestinal endoparasites in sheep. Okay, here we have the results of different concentrations of ethanolic extract and including they quantified the artemisinin content. We can see that the egg hatchability is decreasing when the concentration of the extract is bigger. Okay, who did this work? Amanda did this work. Amanda, she is veterinary, and she found a potential to use Artemisia against endoparasites in sheep. And here we have the potential of this elite Artemisia in the control of Leishmania infantum. Okay, look, the elite Artemisia annua was more effective against intracellular forms of Leishmania than against extracellular ones. We can see the concentration is lower for intracellular Leishmania. It means the artemisinin can cross the membrane. It's so good, it's very good, because the promastigote is the Leishmania inside the mosquito, okay? And a mastigote is the Leishmania inside the animal. This elite Artemisia showed lower toxicity to host cells compared to the parasite. Excellent. The treatment reduced infection indicators in a statistically significant way. Very good. Overall, the compound demonstrates a promising balance between activity and safety. Okay, although further studies are needed to confirm its potential. It's a very, very good result.
Hello, my name is Bruno Henrique Fontoya. My work is about nanoparticles made from elite genotypes of Artemisia annua studied as anti-tumor agents in skin melanoma cells. Cutaneous melanoma is the most aggressive type of skin cancer. It goes deep into the skin, spreads to other organs, and can cause death. Artemisia annua is a medicinal plant rich in phenolic compounds and sesquiterpenes like artemisinin. These compounds have antimalarial, antimicrobial, anti-inflammatory, antioxidant, antiparasitic, antidiabetic, and anti-tumor actives. To make these effects strong for human health, we used nanotechnology to carry plant extracts and nanoparticles. Our results showed lower cell viability and decrease of reactive oxygen species such as ROS and NOx, and decrease of cell defenses like protein and non-protein choice. And this image, we see the fluorescence of tumor cells. Blue shows the nucleus marked with DUP, and red shows the mitochondria marked with TMRE and at the higher concentration. At fluorescence, decrease in minimum mitochondria, damage, and cell death. In conclusion, Artemisia nanoparticles show strong potential as therapeutic agents against metastatic cutaneous melanoma. Thank you very much.
One possibility, another possibility to use Artemisia is doing tablets. Here I would like to honor Dr. Pamela Weathers because she gave a lot of time doing this research. And here in Brazil, we try to do the same, trying to do these dried leaf tablets to treat malaria, including we have a startup company trying to do it, but this company is passing for some difficult time due to the lack of investors. But this is another opportunity to use Artemisia annua. We also is sharing these genotypes, these elite genotypes, and sending it to countries in Africa. We did an agreement with the agronomy faculty of Angola, and we are trying to keep this collaboration. We are finding some difficulties, but we are keeping our effort to find to reach the objectives of this agreement. In the beginning of the work with Artemisia, we also tried to test these Artemisia annua plants as a herbicide, and we could find good results with allelopathy experiments. It was the master thesis of Emanuele Maggiero, Dr. Emanuele Maggiero, okay? And she tested the Artemisia annua aqueous extract, and we found good results in the highest concentration. And thank you. Here you can find my email address, my number. It's also WhatsApp number. And thank you all, and thank you to my group.